Now that we have discussed the studies of morphological plasticity in awake behaving mice, let's move on to the functional imaging studies. In addition to the morphological readouts that I've mentioned previously, such as the dendritic spine turnover, we can also do functional analysis of the synaptic signaling, of neuronal signaling in the brain of awake mice. And again, it's important to study the functional signals in awake mice as opposed to anesthetized, because anesthesia does affect the calcium signaling very strongly. I call this calcium signaling because we use a surrogate readout to image the neuronal activity. Neurons primarily communicate with each other through electrochemical synaptic transmission, but with every action potential that is fired by a neuron, there is a strong increase in calcium, intracellular calcium, which can be detected as an increase in fluorescence of a fluorescent protein that is specifically modified to be sensitive to intracellular calcium concentration. Here is an example from our earlier work. 5-6 years ago, we published this paper where we used for the first time the flat-floored airlifted mobile home cage platform that we now use in our facility on a daily basis. And here you can see a cross-section of a fluorescent signal in the cortex of an awake mouse, and the B part shows the transient increases in the intracellular calcium that are associated with individual neurons firing in the brain of an awake mouse. Now, 6 years ago, when we first started this work, the indicator that was available was GCAMP3, which was not very sensitive, and its dynamic properties were not optimal. Therefore, you can see that the signals here are barely detectable. More recently, we've started using GCAMP6, a more advanced indicator, and you can appreciate for yourself how detailed is the signaling that we can record using this indicator. The movie on the left is taken during the Z scan, so from the surface of the optical section stack to the depth, towards the depth of the stack, and neurons are signaling as we scan them, so it's literally they fire as we go. And you can see that we can measure not only somatic increase in the cell bodies of the neurons, but also we can get many details about the subcellular localization of the calcium increases in the dendrites and axons of individual neurons. And simultaneously with the fluorescence imaging movie on the left, we recorded just a video camera with the mouse moving in the mobile home cage that is shown on the right. Because the imaging was done in the motor cortex, we were able to correlate the movements of the mouse, the motor activity of the whole organism, with the signaling of individual neurons, subcellular structures, and neuronal networks that were simultaneously recorded in the brain of this mouse. Here are some more examples of neurons firing in the mouse brain while the mouse is moving. And on the right hand side you can also see the reconstruction, the projection of the whole cortical columns of neurons imaged from the side, all the way from corpus callosum through layer 6 to layer 1. And here is another beautiful example. They're quite mesmerizing to just watch. So far we've learned what kind of equipment can be used for the studies of neurons and cognition in the same experiment. And I've shown you a couple of readouts, the morphological analysis of neuronal plasticity and also the functional analysis of neuronal signaling at individual cells and the network level. And now I would like to tell you about a project, an exciting project that we are currently starting at the ARB facility with my group that's called the Conscious Evolution Research Group. As you can see, the title of this project has several important aspects to it. The title is Translational Readaptive Plasticity and Mechanisms of Psychedelic Treatment in Rodent Models of Maladaptive States. And there are several important aspects here. The first one is translational. Now, there are ongoing clinical trials in the last 10 years or so with the psychedelic substances such as LSD and psilocybin, which show very promising and strong, powerful results in, for example, models of depression, addiction, and also post-traumatic stress disorder. And all of these disorders can be grouped into the maladaptive states. And by maladaptive, I would need to explain this. I mean that the adaptation, the plasticity can take you to adaptive states where you fit the environment well and you function in the environment quite well. Or if the plasticity goes wrong, then the state can consolidate in a maladaptive state. And these are typical for the disease states such as depression, anxiety, and other mood disorders. And another reason why I'm excited about this project is because we're using the rodent models in which we can test the levels of synaptic plasticity at different organizational levels. In the whole organism, looking at the behavior of the mouse, in neuronal circuits, cortex-wide or in specific cortical areas, at the level of individual neurons, at the level of individual synapses. And because the psychedelic drugs have a very specific target, the serotonin type 2A receptor, 5-HT2A, we can also study it at the molecular level using the agonists and antagonists and receptor expression in specific neuronal populations. And all levels can be assessed at the same time. As I mentioned, the animal is awake and behaving during the experiment. The fluorescent indicators allow us to image the neuronal activity at the level of individual neurons or networks at the same time as testing behavior. We can also look at morphological plasticity using dendritic spine turnover. And the only step that does require sacrificing the animal at the very end of the experiment is the step where we do biochemical and molecular biological analysis of the protein levels, protein expression, phosphorylation, etc. The hypothesis that we are testing is that in the maladaptive states there is still a potential for readaptation if a certain drug, for example a psychedelic or antidepressant drug, and many other drugs that are increasing plasticity, if the certain drug is used in combination with the right therapeutic setting, in which case then the readaptive plasticity occurs and the organism, both psychologically and physiologically, returns to the road that it left previously during the maladaptation. Our approach in this project is to study plasticity at several different levels at the same time. So we're actually talking about the interconnected plastic changes like feedback loops that go between the cortical level, the layer-specific, cell-specific, synapse-specific, and molecular level of plastic changes. So we are kind of attempting to study metaplasticity, if you will, although this is not a very common term, but we like to think of our study as a metaplasticity study. As I already mentioned, the objective of the study is to provide mechanistic understanding of the psychedelic therapy. And the mechanistic understanding of this kind cannot possibly be obtained in those human clinical trials that are ongoing now in the Imperial College, for example, at Johns Hopkins and New York University and several other places. Those are clinical trials that are very promising and very important. They use human imaging, non-invasive imaging techniques, and behavioral techniques, but they cannot, for ethical reasons, penetrate down to the levels of individual cells, cellular networks, receptors, and dendritic spines. Because psychedelic drugs have been stigmatized and prohibited, classified for 50 years, it's a very interesting situation with them. This area has been severely understudied compared to other similar drug classes, and it's an enormous opportunity right now to look at the mechanisms of action of these drugs using the techniques and methods that were developed in the adjacent areas with legal drugs such as prescription antidepressants, for example. The specific hypothesis that we want to test is that the psychedelic drug treatment produces a shift in the activity mode of the layer 5 pyramidal cortical neurons, the very same neurons that I showed you in those beautiful yellow fluorescent protein expression images before. And the layer 5 pyramidal cortical neurons are known to express high levels of 5-HT2A receptor, therefore they are considered a major target for the psychedelic treatments. And as you can see, our hypothesis links the neuronal level with the network level and the whole organism level. So it really is the neuron and cognition in one experiment. And this is why the ARP facility, the Awake Rodent Research Platform, is so strategically well situated to study this, because we do have the equipment and methods and the expertise to study neurons, synapses, networks, and behavior at the same time in one experiment, and correlate between these different modalities and levels, and also possibly start finding some causal relationships, not just correlations. This is a busy slide, but it shows the workflow of our experiments as we have planned them. In the A panel, you can see that we will be using 2 strains of mice that steadily express the reporter proteins, fluorescent proteins that show either the shape of the neurons or the level of intracellular calcium. They will be subdivided into 2 groups. One will undergo no specific stressful treatment, and the other one will be injected with the stress hormones, the corticosteroids. And this will produce a certain behavioral phenotype which will be characterized in all these mice in the C panel for the readouts, phenotypical readouts typical of different mood disorders, maladaptive states, such as depression, for example. We would use the sucrose preference, whether the mouse prefers the sweet water over non-sweet water. We will check the weight of the mice. We will study them for place preference, whether they prefer dark places or light, to stay in the light. We will check their ability to recognize the novel, pay attention to the novel objects, and also the sociability tests, as I showed you in the previous slides. We will also test these mice for hyperalgesia, which is known to be associated with a chronic stress treatment. So their paws become extremely sensitive to mechanical and thermal stimuli, and we will test this phenomenon that is called allodynia. Another phenotype that is associated in mice with a chronic stress model is the susceptibility to alcohol addiction, for example. So we will test, and this is also a translational readout, because many of the depressed patients have a tendency to become alcoholics. After the behavioral phenotyping is completed, we will have a baseline before the treatment with either LSD or psilocybin, where we will use 2-photon microscope to image the spine turnover simultaneously with behavioral tasks. And in the second strain of mice, we will do calcium imaging at 2 different scales. One is the whole cortex scale with wide field imaging with behavior, and another one zoomed in with a 2-photon to look at individual neurons in layer 5. Once this baseline is acquired, we will then image the mice throughout the treatment session, and we will test the changes, acute changes that will be induced by the drug. And then the mice will be left back in the cage, and the next day and the day after and the week after and the month later, they will be brought back for another imaging session in which both the behavior and the brain signaling and brain morphology changes will be characterized. And finally, in the G panel, at the end point when the mice will be terminated, their samples will be collected and analyzed using biochemical and molecular biological techniques. To summarize this part, many leading groups around the world today are trying to combine different methods in order to bring together different levels or scales of organization of the nervous system. From in vitro, dissociated cells and single receptors and molecules, to in vivo experiments, and finally to the in vigilo experiments where the animal is awake and vigilant and behavior is measured and assessed at the same time with the brain signaling, bringing neurons and cognitive function to the same experiment. And of course, importantly, we try to keep in mind that these experiments are primarily designed to learn something about the human nervous system so we can become more adaptive to the changing world. So in Homo experiments are also kept in mind, and we try to keep our collaborations with the groups working in humans, such as Robin Carhart Harris's group and David Nutt's group at the Imperial College, and also with companies that are developing novel therapies and drugs, so that we always keep in mind the human aspect of these translational experiments.